The fast detection strategy allows the early detection of cyanobacterial blooms and related cyanotoxin, either in water samples, either in organic matrix, such as shellfish and other fish products.
Cyanobacterial blooms are due to the overgrowth of cyanobacteria, which can live in any kind of environments and have emerged as an environmental problem across the globe within the last 15 years.
As the number of harmful cyanobacterial blooms has increased during the last several years, the need for their early detection, which is key for addressing bloom and toxin spread, has become more urgent.
Our fast detection strategy combines remote and proximal sensing technique and technology with the laboratory chemical and bioinformatic analysis in a unique integrated workflow.
The whole process is safe. Appropriate safety measures are taken to prevent aerosol inhalation and skin contact during sampling and lab analysis.
For data retrieval, first locate the target area on a global world map and retrieve the data from various public and private remote sensing data sets for the collection date. After retrieval, process the raw data, calculate the multispectral indexes, and classify the resulting information. Then define the sampling sites on the generated thematic map. For sample collection, transport the equipment to the selected sampling site in the mobile lab and plan the drone flight path for performing a macro area survey. At the site, use several drones.